Hello, can somebody please confirm you can hear me?
I can hear you.
Thank you. And shut that down. Shut that down. Sorry for being late. I was checking through the final to make sure that everything I was going to tell you was correct. And I noticed that I've got two questions from Lab 8. So I was going to tell you that Lab 8 was not on the final and not on the quizzes, but actually there are two questions from Lab 8 on the final. So everything is now on the final. Any questions about any of that?
Wondering when we're going to drop the lowest score quiz just to see where our grades are at.
Yeah, I've been trying to get the infectious disease project graded so I can put up the scores. And I still have four people to grade, so that's what I've been putting my all my effort in. I hope to be finished sometime late tonight, probably early tomorrow morning, and put up your score for the infectious disease project. Once I get that done, I will then grade the, well, I don't need to really grade it. I just need to figure out which quiz to drop and change quiz 5 so it's worth 75 points. So that's my second project. Any question about any of that? All right, sorry, the infectious disease project takes me a lot of time to grade, and so I've got most of the class graded, but I won't put up the grades until I have everybody graded. And I've still got four students, actually three and a half. One of the students I got started on last night. Um, all right, let me call up the syllabus. I want to leave that there. Let me state that all lectures and labs will have a question, at least one question. Why isn't that showing? Oh, there it is. Uh, except for chapter 2, there is no direct question on chapter 2 because we have to understand chemistry to answer some of the questions, but there's no question directly from chapter 2. All other chapters and all other labs will have at least one question in the final. I don't think you're seeing my screen. Let me show that. There we go. There we go. However, the majority of the final will be on the material since the last quiz, meaning the majority of the final will be covering chapter 7, 13, 14, and lab module 9. There are two questions from lab 8, but that's it. And it's not stated here, but there are a few questions from the unknown project on the final too. Now, the unknown project does have some case studies, which you had actually in a number of the labs. And some of you are kind of weak on doing case studies, so you may want to, I don't know, practice that. If you missed any of the case studies, you probably want to practice the case studies because there's a number of case studies questions. Some of them are pretty simple and they'll be in the multiple choice, but some of them are difficult and they'll take you, I don't know, several minutes at least to finish, and they will not be in the multiple choice final. there is a final A. Let's see if I can get there. Am I in the wrong place here?
Or something like that when it's gonna take a few minutes. Are we gonna be allowed
That's
to have?
it's final A. There's probably only three questions, but they will be lengthy. I think I've given you 1 hour on that to take.
May we may we be able to have scrap paper to kind of start to write down our thoughts and keep track of things?
You may have scrap paper, but you have to show me the scrap paper so I know that it's blank paper. So if you
Thank
have
you.
scrap paper, make sure you show it to me before you start writing on it. Um, but short answer questions, it is time, but must be completed in the designated time. I think I give you, nope, I only give you 45 minutes because nobody last term took anywhere near an hour. But the point is, you will be what photographed or making a movie about it, so you can't cheat on it. So don't try to look up the answer. Don't try to use AI to get the answer because it will be recorded. And there's only three questions, and it probably won't take you anywhere near 45 minutes. I think the longest somebody took last term was something like 15 minutes. But you do have 45 minutes, and they are detailed questions where you are going to have to work them out. And that's why I'm giving you 45 minutes. There's only three questions. Um, it's not really an essay type question. It's more like your unknown project where you're given a bacteria and you're given different results. You have to look up the pictures of the results and then figure out what those results mean. Look up a dichotom is key, and then identify your unknown. So it's a very detailed case study, is what it is. Any questions about any of that? And by the way, I should briefly mention, although this is supposed to be a review of the final, I should briefly mention that um, wrong place. That the unknown project, the last time you can request a test is Friday. If you request a test on Friday, it must be a test that you can get the results in 1 day or less. And you do need to finish your unknown notes pages. They are worth 5 points. That counts as a full lab. A lot of you have not finished your unknown notes pages. Any question about any of that? All
Yeah,
right.
so like I finished my requesting tests and the information I gave you on that, is that the 5 points or is there something else I need to do for that aside from the report part?
Well, it tells you in the unknown notes what it should be in the unknown notes. If I've given you a comment that you've finished requesting tests, that means everything is almost correct. You just have to make sure that you have everything in the unknown notes that should be there. You should have the results from the um, one or two emails that I sent you. You should have the conclusions from those results. You should have your test request, which will obviously be there. And you should have the results that I sent you and your conclusions. Now, remember, a result is an observation. The tube was yellow. The conclusion is what's the conclusion of that yellow tube, and that is the test might be positive. Okay? So make a distinction between the results and the conclusion, and then you should put in the species currently under consideration. So that should be there for every test you request, species under consideration. And then everything else should be in there that's related to the unknown notes page, but I'm not going to require it to be there. You don't need to put a date in there because the unknown notes does put a date in there automatically. When you copy your unknown notes pages into the unknown report, if you write your unknown notes formally, you can directly copy it into your unknown notes, not your unknown report. However, if it's not formal, then you should make it formal for your unknown report. And you will need to add the date for the thing you're copying because you can't copy the date. Any question about that? All right. Oops. What did I do there? Not sure. Are you still seeing me? I have the share still going, so that's good. All right. Um, so once again, the majority of the questions from the final will be covering chapter 7, 13, 14, and lab module 9. There are 2 questions from lab module 8, which wasn't in the quizzes. And then all of the other labs and chapters, except for chapter 2, have at least 1 question. None of them have more than 3. I think like chapter 8 might have 3, except for chapter 1. Chapter 1 introduces a lot of topics. It also introduces the history of microbiology. Do you think I like the history of microbiology? That's a yes or no question.
You love it.
Yeah, I love it. Okay. Um, well, the point is, I do like it, and so I tend to ask a little more questions from chapter 1 because I like the history of microbiology. In the history of microbiology, you never need to know the dates, except for the very first slide where I showed you when we have the fossil record for the first life on the planet Earth. So you need to know that date, but all the other history of microbiology, you don't need to know the dates, but you should know who was responsible for the finding and then what the finding was in the history of microbiology. Like Dr. Koch was responsible for discovering the first bacteria that was causing a given infective disease, anthrax. And then he also wrote up his procedures for doing that, and that was known as Koch's postulates. And Louis Pasteur was the first person to obviously start pasteurization. He did a lot of things that were the first, like he was the first one who figured out the vaccine, how a vaccine works, and how to make vaccines. And he also the first one to come up with aseptic technique, where you would use aseptic technique to get a species purified so that there'd only be one species growing in the container, the media, and then he would use aseptic technique to keep it pure, keep it sterile, meaning it would always have that one infective species in it. All right, any question about any of that? If not, let's begin with the last. There are questions about the unknown project on there, and they're kind of like case studies. So you're just going to have to live with those. And some of them are detailed. There's like 2 of them on the, what is it, the final A. Lab 9. Let's talk briefly about Lab 9. Identification of gram-negative bacilli. There are two gram-negative bacilli that this chapter is talking about, and that is the Enterobacteriaceae and the Pseudomonaceae. So you should know how to separate those two families because they're both gram-negative bacilli. We also talked about the Imvik reaction, so know what the indole test is, the methyl red test, the Voges Perskira, the VP test, and the Simmons citrate test. We talked about carbohydrate fermentation here, so you should know about that. And this is like the second time we talked about it in the second lab. Motility, we talked about the motility test, and then hydrogen sulfide production test. Realize there are three different tests you can use to test whether there's hydrogen sulfide being produced. If hydrogen sulfide is produced in any of these three tests, you'll get a black precipitate forming. And the three tests that you can use to determine hydrogen sulfide production are triple sugar iron, or TSI test, or the SIMS test, the sulfide indole motility test, and then growth on HE or hectone enteric agar plates. And if there's hydrogen sulfide being produced, you'll always get black. Any question about any of that? All right, you know the Enterobacteriaceae is a very large family, 53 genera, and 24 of those genera are known to cause human infections. Some of them are frank human pathogens like Salmonella, Shigella, and then E. coli, Serovar 015787. E. coli itself does not normally cause disease when it's staying in the intestines or the colon, but 015787, this strain, will cause human disease. Pseudomonas aeruginosa is the main member of the Pseudomonas family or the Pseudomonaceae family that cause human disease. And a simple test to separate the Pseudomonaceae family from the Enterobacteriaceae family is the oxidase test. The Pseudomonaceae family is oxidase positive. The Enterobacteriaceae family is oxidase negative. However, there are other ways you could do it. The Enterobacteriaceae family, all of the members of the family ferment the sugar glucose. None of the members of the Pseudomonaceae family ferment the sugar glucose. The Pseudomonaceae family, all of the members are aerobes. And then the Enterobacteriaceae family, all of the members of the family are facultative anaerobes. All right, you should know that the indole test positive result is, it's kind of purple here, but it should be reddish. And then the UMAR test, if it's positive, it's reddish. The VP test, if it's positive, oops, right here, it's reddish. The entire tube is not red, but the portion with the supernatant is. The stuff you add to it is red. Simmons citrate test, you should know if the tube turns blue and you see growth on the tube, then it's positive. No growth and the tube stays green, it's negative. The motility test, if the growth is only in the stab, the organism is not motile. If it flares out of the stab, it's slightly motile. And if it's highly motile, you'll see that the growth is all over the tube, and so the tube will look cloudy, like this one here. For the carbohydrate fermentation test, you have one sugar in the tube, and you could call it the glucose fermentation test or the lactose fermentation test or whatever the sugar is. And I just call it the carbohydrate fermentation test to use one name for all the different sugars. If it's positive, then it will be A, and there's no gas being produced for A for acid, and the tube will be yellow. If it's A and gas being produced, acid and gas, and the tube is yellow, and you see a bubble in the inverted Durham tube, and that's AG. For negative, the tube will remain reddish or pinkish. I already talked about hydrogen sulfide production. You'll see black someplace.
You were just on talking about how positives is A. Would the negative that was above be K?
Yeah, you could call that K as well as negative.
Gotcha. Okay.
Okay. And you really don't call that positive because AG and then A are both positive. So you really have to say A or AG. And I would obviously have that in the multiple choice, either A or AG as a possibility. For the triple sugar iron test, it's a complicated test, and I don't know an easy way to memorize this. For gas production, you'll see cracking of the media, and there will be a gas bubble. No gas, no gas, no gas. For hydrogen sulfide being produced in triple sugar iron, they'll be black, especially on the lower portion of the tube. For the glucose and one of the other sugars, either sucrose or lactose being fermented, both the slant and the bottom of the tube, the butt, will be yellow. If only glucose is fermented, the bottom will be yellow because the sugar is fermented slower rate anaerobically than aerobically, and then the slant will be reddish or pinkish because all of the sugar has been used up. And rather than the bacteria start to die, it starts fermenting or using the proteins. I should say using because utilizing, not fermenting the protein. And then that makes a base, the ammonia, and that changes the color from yellow to pink. Now, in the time we look at it, the slant would be pink because we look at it after 18 hours. But if you were to look at it at some time earlier than that, let's say 9 hours or 12 hours, that slant would actually be yellow. Any question about any of that? And then the negative, also called the K over the K, would be a pink slant or reddish slant and a pink or reddish butt. For the SIMS test, 3 tests are going on at once. Sulfide production, which will turn black. Indole production, you add Kovacs reagent, and then it'll be pink where the Kovacs reagent, which floats above the liquid, so it will be pink. If it's negative for indole, it'll be yellow. And then you can look for motility. There you can see a stab. Maybe you don't see it too well, but the growth is only in the stab. And there, the growth is flaring out from the stab right there. And so that is motile. This one is not motile. All right, any question about Lab 9? So let's move on to briefly Lab 8. Oh, I already got that open. So lab 8 is looking at the gram-positive cocci mentioned here and the gram-positive bacilli mentioned here. I don't think I'm asking any questions specifically about these, although there could be a case study about, Staphylococcus aureus or something like that, but no question directly on the lab concerning these families. On this lab, it's more important to understand the CAMP test, where we're looking at the synergism between beta hemolytic organisms growing near Staphylococcus aureus, which is also beta hemolytic. And in the CAMP test, if it's negative, there is beta hemolysis happening by Streptococcus pyogenes, but it's normal. I have to blow this way up where you can really see it. So the dark pink string is the bacteria, and then the clearer area around it is the beta hemolysis. And then the Staphylococcus aureus, the dark, I don't know, orange color here, that's the bacteria. The clear region, or the clearer region, is the beta hemolysis from Staphylococcus aureus. Streptococcus pyogenes is CAMP negative. There's no enhanced hemolysis in this region of the Streptococcus pyogenes streak. Listeria is slightly enhanced. We have a very minor enhancement of the hemolysis right here, a very minor arrow. And then Streptococcus galactiae is a enhanced hemolysis of the hemolysis in the region of Streptococcus galactiae grown near Staphylococcus aureus, meaning you see a very clear arrow. Any question about that? Let me draw that down. Biolesculin test, if the esculin is hydrolyzed to glucose in esculatin, you get a dark brown color. It's actually, it looks black. And biolesculin negative, it's either very minor darkening or it's no darkening at all. And then the Streptococcus faecalis broth test, called the SF broth test. If the organism can ferment glucose, it will turn yellowish color, positive. If it cannot, it will stay purple in color. And then the optogen sensitivity test. If the bacteria is sensitive to the antibiotic optogen, it will have a zone of inhibition greater than or equal to 14 millimeters. If it is sensitive, this is sensitive to the antibiotic optogen. If it's resistant to optogen, then the zone of inhibition will be less than 14 millimeters. This is showing you there's no zone of inhibition, which is less than 14 millimeters. You should know how to use a dichotomous key. All right, any question about Lab 8? If not, let's go on to Chapter 7. Obviously know the terms. In fact, you should really know all of the objectives, which you can find in the study guide for Chapter 7. You should be able to discuss the factors that influence microbial death rates. I'll get to that in a minute. Be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells. you damage the membranes, like the cell membrane. You can damage or inactivate, or what's the term? Drawing a blank there. Damage or inactivate proteins. And the third is damage DNA. Those are the three main ways which antimicrobial agents kill or inhibit cells. I can't remember the term for proteins. Inactivating a protein is changing the shape of the protein, which I forget. Anyways, understand the various types of disinfectants and how they work, and understand the relative resistance of the major microbial groups to antimicrobial agents. You should know that microbes die at a constant logarithmic rate, and that the rate of microbial death will depend on the number of microbes. The more microbes there are, the longer you need to expose the microbes to the agent, like the antimicrobial agent. The time of exposure is obviously important. You have longer times of exposure if you have endospores present. And then microbial characteristics can affect the microbial death rate. Like one example would be the family Mycobacterium. They have mycolic acid in their cell wall, and so that can affect the microbial death rate. If you have a antibiotic that's water soluble, or an antimicrobial agent which kills and it's water soluble, it's probably not going to get into, or at least not get into, the Mycobacterium very well, because the mycolic acid will keep it from getting into the cell. And then lastly, there can be environmental influences affecting the microbial death rate. Organic matter can inactivate the microbial agent, like bleach is very easily inactivated by organic matter. Most hospital specimens are around organic matter. And then the temperature can affect the microbial death rate, as well as the presence of a biofilm. We didn't really talk much about biofilms, but it's something that some organisms put out, which then can coat the microbe in the biofilm, and then the biofilm can help the microbe survive harsh environmental conditions, such as antibiotic treatment, for example. Any question about any of that? You should know the terms. I'm not going to skip over them, just saying you should know the terms. I don't have the word there. When you coagulate a protein, what is that called? Nobody's getting it either. I'm just drawing a blank on what that's called. You inactivate the protein. It'll come to me. All right, you know the physical methods for controlling microbial agents. You should know about pasteurization. There's two processes for pasteurizing something like milk. You can expose it to 30 minutes for 63 degrees Celsius, or you could expose it to 15 seconds at 72 degrees Celsius. Pasteurization is not ultra high temperature milk. This will actually sterilize the milk, where you heat it for at least 1 second at 140 degrees Celsius. Dry heat, wet heat filtration, low temperature. It does not kill cells necessarily, but it does prevent them from producing toxins, growing. That's not true of an agent growing in your refrigerator, but it is true of most microbes, like most human pathogens. High pressure, desiccation, radiation, you should know all these. Chemical methods of achieving microbial control. I did mention most chemicals do not kill all microbes. There's only a few chemicals that actually kill endospores. Hydrogen peroxide and other peroxygens are one chemical that does kill endospores, but many chemicals like bleach do not kill all endospores if they kill any endospores of the species. Bleach, some endospores can survive. Some endospores are sensitive to bleach, but the point is that there are some endospores that bleach would not kill. You know the term disinfection. You know the types of disinfectants. All right, and then the microbial characteristics. Microbes have different characteristics affecting the resistance of the microbe to the antimicrobial agent. Prions are about the most resistant microbe we have. The only way to really remove prions are to incinerate it at like 1,000 degrees. I forget if that's Fahrenheit or Celsius. It doesn't matter. It's very, very hot. Incinerate the prion and that destroys it. The other way you can destroy prions are subject it to um, proteases, digesting the prions, and that's something that they use on like surgical instruments, which you don't want to subject them to 1,000 degrees Fahrenheit because that would damage them. Endospores, the next major resistant microbial group. Endospores are very resistant to treatment. They survive many different conditions, whatever you throw at them. Like I said, there are some exceptions, and mainly the peroxygens, the hydrogen peroxide, can kill endospores. Mycobacteria, the next most resistant, although this characteristic, although it makes it resistant to any antimicrobial agent that dissolves in water, it is very sensitive to an antibiotic which is fat soluble, like, oh gosh, what is that called? Streptomycin, there it is. Took me a while to figure it out. So it is sensitive to streptomycin because streptomycin is fat or lipid soluble. It goes right through the mycolic acid of the mycobacteria. And then alcohol. Mycobacteria are very sensitive to alcohol, even though mycobacteria are not sensitive to most other antimicrobial agents that are water soluble, at least. Cystiprotozoa, vegetative protozoa, next most gram-negative bacteria are more resistant than gram-positive bacteria. The gram-negative outer membrane can keep certain agents away from the bacteria. The only one I mentioned was penicillin G. Cannot get through the outer membrane of the gram-negative cell wall. Viruses without envelopes are more resistant than viruses with a lipid envelope. Gram-positive bacteria, one of the most sensitive microbe to any antimicrobial agent. And then the most sensitive thing are viruses with the lipid envelopes. Any question about any of that? All right. That's it for chapter 7. Let's move on to chapter 14. Oh, wait a minute. Let me make sure chapter 14 is One of the ones, yes, it is. All right. Know the terms of this chapter. You know the terms of this chapter. You've learned 50% of this chapter. Understand the relationship between the normal microbiota and its host. Understand the stages of disease development. And then lastly, understand what nosocomial infections are and how to control them. If you go out into the healthcare industry and work, you should know what nosocomial infections are and how to control and prevent them. One of the best ways is simply to wash your hands. After you touch a patient, you should wash your hand before going on to touching another patient. Frequent hand washing reduces nosocomial infections by about 50%. And that's true of healthcare workers as well as visitors coming into the place. Nosocomial infections are an infection the patient did not have until they checked themselves into the healthcare facility, meaning they pick up that infection while they're in the healthcare facility. You should know that everyone has normal microbiota, a large community of microbes found in association with a person. And there are communities with all animals and plants, and it'd be the normal microbiota of that animal or that plant. For example, the normal microbiota of cats is slightly different than the normal microbiota of humans. Humans do not have Bartonella henselae as a normal microbiota, but cats do. And when a person gets Bartonella henselae, it causes cat scratch fever in the person. So something which doesn't cause much disease in cats, part of the normal microbiota, can cause disease in humans because it's not part of our normal microbiota. The normal microbiota is only found in certain locations, and if it's found outside of that location, it can cause disease, like E. coli growing in the ear, can cause an earache or an ear infection, growing in the lungs, causing pneumonia, but grown in the intestines. That's where it's normally supposed to be. That's part of our normal microbiota. Showing you the locations of the normal microbiota. Normal microbiota is different from the transient microbiota because the transient microbiota is something that you can find growing on the person for days, weeks, or months. The normal microbiota generally will, once it colonizes the patient, will remain with the patient pretty much for their entire lifespan. Like most of us when we were born, we got Staphylococcus epidermidis from probably our parents when we were born, and it is now a part of our normal microbiota, and we'll have it until the day we die. A transient microbiota, fortunately, I had Staphylococcus aureus growing on my skin transiently after I hugged my aunt, who she actually hugged me, and she had a Staphylococcus aureus skin infection. She probably thought, oh, I'm not contagious because I'm taking antibiotics, but I did get it. And fortunately, it was transient. The next time I tested myself 3 months later, I had no more Staphylococcus aureus on my skin. Any question about any of that? You should know we have more bacteria cells in and on our body than we have normal human body cells. You should know about microbial antagonism, the competition between microbes and our normal microbiota can compete with a human pathogen, either occupying the niche, competing for resources, so there's no resources for the human pathogen to then grow, producing acid, making the environment less friendly for the human pathogen, and our normal microbiota do that in our mouth and in our skin and in the urethra, making acid so that some human pathogens cannot get established. And then there are some of our normal microbiota, like E. coli, which actually produce a bacteriocin, a protein that has a narrow acting, what do you call narrow acting ability. So it's only going to knock out the E. coli producing bacteriocin, will only knock out the human pathogens Salmonella and Shigella. And so if you get Salmonella landing right next to this E. coli making the bacteriocin, that Salmonella will not get established in your gut and will actually be killed. And you can think of a bacteriocin as a very narrow acting antibiotic. It's much more narrow than any antibiotic, but it is similar to an antibiotic. Any question about any of that? All right, you know about symbiosis, two different organisms or populations living together. That symbiosis could be mutualism, benefit to both populations. Commensalist, where one organism benefits, but it doesn't harm the other. And then parasitism, where one organism harms the other. Human pathogens are parasitic in the sense of parasitic symbiosis, meaning living with us and harming us. Some of our normal microbiota are mutualists, like E. coli, helping us digest our food. We give E. coli food in a home, so it's benefit to them, so it's a mutualist. And then the best example of a commensalism is where we have a nematode in our eye, one in each eye, which we provide a home for the nematode, and the nematode doesn't do any damage to us. I'm assuming the nematode was eating bacteria that falls into the eye. Any question about any of that? So the nematode doesn't do any harm to us, and we provide a home for the nematode, commensalistic. So, an opportunistic organism is a part of the normal microbiota that can cause disease in certain situations. Normally, the normal microbiota does not cause disease, but individuals with compromised immune systems, or when the normal microbiota grows in a location it should not be, it can become opportunistic and then cause disease. You should know about Koch's postulates. We talked about it in 3 different places in this class. This being one chapter here, probably the last time we talked about Koch's postulate. The first time was chapter 1, and I don't remember the second time, but Koch's postulates are the steps used to identify a given organism causing a given disease. So you get a diseased individual, get an organism out of it, grow it up and in tissue culture, purify it, make sure you have a pure culture of that one species, then inject that one species into an organism. If the individual becomes or organism becomes ill, and then all cases of this individual that are ill have that one organism. And then furthermore, Koch's postulate states out of that ill organism, you must be able to get the organism that you injected into it. If Koch's postulate is met, we then show that this organism causes this disease. All cases of this disease must be caused by this organism. Any question about any of that? Well, there are some exceptions to Koch's postulate because there are some microbes with unique culture requirements that cannot be grown in tissue culture. For over 100 years, people were trying to get the organism that causes syphilis to grow in tissue culture. And until recently, the last 5 or 7 years, they never figure out what you need to add to tissue culture to get syphilis to grow. So for a large number of years, syphilis was an exception. We knew that syphilis, the organism, caused that disease, but they couldn't grow it up in tissue culture, so you couldn't do Koch's postulates. And then what do you do if there's multiple organisms that cause the same disease? Same signs and symptoms. As I mentioned, there's more than 3 different species that a virus that causes the human colds. There's actually 3 families of viruses that cause the human colds. And in Koch's postulate, you have to have one species causing one disease. And then there's moral and ethical conditions for human-only diseases. Like nobody's injected people with HIV and then later followed them to determine that they developed AIDS because of moral and ethical reasons. But we know that HIV causes AIDS because we've done large epidemiological studies. And then we follow those people over time. And some of the HIV negative people became HIV positive. And then we followed those people over time. And then some of those HIV positive people then went on to develop AIDS. Any question about any of that? You should know the difference between a sign and a symptom. You should know the terms. I'm just going to say know the terms for this chapter. You should know about herd immunity. You should know about patterns of disease development, predisposing factors, the development of disease. Oops. Come on. Most diseases follow the same development of the disease. There's an incubation period when the person first becomes exposed to the agent causing disease. The incubation period lasts until the patient has signs and symptoms. When the patient first has signs and symptoms, if they're mild, we call it the prodromal period. Like when you first come down with a sore throat and you say, geez, am I coming down with a sore throat? And you have to think about it. And you say, yeah, I've got a slight sore throat. That's the prodromal period. The period of illness is where you don't have to think about it anymore. You say, oh, geez, I've got a burning sore throat. When the person is most ill is in the period of illness. It's also typically when the patient has the most number of microbes. That's followed by the period of decline, and the period of decline begins when the patient starts feeling better. The patient has a fever, they're in the period of illness. The fever breaks, that starts the period of decline. The period of decline continues until the patient has no longer any signs or symptoms of the disease. Once the patient has no longer signs or symptoms of disease, the patient enters the period of convalescence. The period of convalescence continues until the patient throws off all the disease-carrying organisms. Any question about any of that? Now, not all diseases have all the periods of disease. Sometimes the flu has no prodromal period. You'll be walking along fine, and then the next minute it feels like somebody hit you with a sledgehammer. So it moved immediately from the incubation period to the period of illness. And then diseases which kill the patient, there will be no period of decline, no period of convalescence. There are some diseases like chronic cystic liver fibrosis, chronic liver disease, what is that called? I forgot. Where there is no real period of decline or period of convalescence, meaning the patient stays in a period of illness and then just might plateau off. They might even decline, get better, but then they'll get worse with time and get better. And the point is there may not be a period decline and there is no period of convalescence. The patient has the disease until the day they die. My mother actually had shingles for 3 years before she died. So she always had shingles. When she got shingles, it never went away. She was a very unlucky individual. And so she never really went into the period of convalescence. There was no period of convalescence, and debatable whether it's a period decline or not. She did get better, so it's like she went to the period decline and then stayed in the period decline. She know about reservoirs of infection, the transmission of disease. She know about nosocomial infections, and that there are extremely important, with between 5 and 15% of all hospital patients or nursing care patients or anyone checking into a healthcare facility coming down with a nosocomial infection. We should know why a patient can get a nosocomial infection and then know the terms. All right, any questions about this chapter? Let's move on to chapter 13. Viruses, viroids, and prions. Know the terms of this chapter. Understand the characteristics, the structure, and the classification of viruses. Understand about viral multiplication. All viruses have a life cycle. With bacteriophages, bacterial viruses, there are only two life cycles, the lytic life cycle and the lysogenic life cycle. With animal viruses, there's main two, but there's at least two others, and that is there's the lytic life cycle, there's the budding life cycle, those are the main two. There's the latent life cycle, that's usually a budding life cycle that then goes latent, the virus goes dormant, is not actively reproducing, and it is not causing disease, latent life cycle. But it can come out of the latent life cycle and then re-enter the budding life cycle and then cause disease. And then there's also the persistent infection life cycle, and it's very similar to the budding life cycle. The difference is the virus is a naked virus. You really don't need to know this because I doubt I ask you a question about the persistent life cycle, but it's a naked virus, and it does leave the cell in a organelle called the vesicle. And the vesicle will fuse with the cell membrane, dumping the virus outside the cell. So it's similar to the budding life cycle, but it doesn't ever get a bud or a membrane bound. Excuse me here. A membrane bound envelope around the virus. Any question about viruses? You know about the host range of a virus, and it's determined by the virus binding the host cell. The virus has certain molecules on the virus that specifically bind to receptors on the host cell. Receptors are just a molecule that the virus binds to, and so there's a specific attachment. Most viruses only bind to one or very few species, and usually just a few cells of that species. Like HIV only binds to, as far as I know, human cells, and only a few cells like T lymphocyte and brain cells and macrophages and a few other cells, but those are the main three. And it doesn't bind to like skin cells and the nose cells and bone cells. And then it can also bind to the cells of the chimpanzee. One of the chimps, I should say, not the other chimp. There are two species of chimps, if you don't know. Do you know the relative sizes of viruses? You should know a virus has a nucleic acid. Its nucleic acid can be either DNA or RNA, never both. And the DNA or the RNA can be double-stranded or single-stranded. Double-stranded RNA is unheard of in our cells, unless the cell has a double-strand RNA virus infection. And then if the genome is single-strand RNA, you do need to state whether it's positive single-strand RNA or a negative single-strand RNA genome. All viruses have a capsid, that's the protein coat surrounding the viral genome. Some viruses have an envelope around the protein coat capsid. Not all viruses do. If it does have an envelope, we call it an enveloped virus. And if it doesn't have the envelope, it is a naked virus. You should know the general morphology of a virus. Helical virus, polyhedral, and then complex. A complex virus can take on any shape. You should know that in viral taxonomy, we don't have much. Let me see, King Peterson came over from, so we do have orders to viruses, and we have families of viruses, but higher than order, we don't really have it for viral classification, with the exception of the kingdom. We do classify viruses into the kingdom that they infect, like there are animal viruses and there are bacterial phages, bacterial viruses that infect either animal cells or bacteria cells. So we do classify viruses into the kingdom they infect, and then into an order. Perhaps all viruses have family. The family name ends in viridae. The genus name ends in virus. And then we have the viral species. And you only use a common English name for the viral species. Like the poliovirus is the common English name for the virus species. And I shouldn't use that as an example because I don't know the genus and the family name. Well, the genus would be the polio virus genus, but the family would be the polio. Actually, the family, I do know that one. It's Picornaviridae for the family. But an easier example would be herpes simplex type 1, which is actually the subspecies. So this the number is the species name herpes simplex. The genus would be the herpes genus virus, herpes virus. The family herpes viridae. Remember, the subspecies is really important in a virus because the subspecies can really determine whether the virus infects the patient or the difference in the different infective, like herpes simplex type 1 tends to cause cold sores. Herpes simplex type 2 tends to cause genital lesions. Another example would be poliovirus type 1 causes the disease polio. Poliovirus type 2 is the oral anti virus vaccine that we use to prevent from getting the disease polio. So that's the Sabin vaccine. Any questions about the subspecies? How important it is? Already talked about that. You should know the 5 different stages of the lytic life cycle of a bacteriophage. You should know the lysogenic life cycle of a bacteriophage. In the lysogenic life cycle, the virus infects the cell, injects its DNA into the cell. Immediately, the virus has to decide, am I going to enter the lysogenic life cycle or the lytic life cycle? If it decides to enter the lytic life cycle, it then replicates the viral genome, replicates the viral capsid proteins and any other viral protein it needs, lyses the cell, releasing the newly released viruses, and that's the lytic life cycle. If, on the other hand, the virus decides to enter the lysogenic life cycle, the viral genome integrates into the host cell chromosome. We call that a prophage for a bacteriophage. And then when that cell divides, both of the cells, daughter cells, will get the prophage. And that's the only time the virus replicates, when the cell replicates. For unknown reasons, the prophage may come out of the chromosome and then not re-enter, but enter the lytic life cycle. And they don't have a good understanding about why the lysogenic virus leaves the lysogenic life cycle and enters the lytic life cycle, other than if you damage the DNA of this cell. Any question about any of that? You should know that if a cell is lysogenic, meaning it has a virus integrated into its DNA, that cell can take on new properties because it has new genes, the genes of the virus. And the best example of that is Corynebacterium diphtherium, only produces the Corynebacterium diphtherium toxin when the Corynebacterium diphtheria is living in harmony with the lysogenic virus. Because it's the virus that codes for the Corynebacterium diphtherium toxin. Any question about any of that? All right, animal viruses generally follow the pattern of bacteriophage multiplication. There are some differences. The main difference is an animal virus can replicate in the cytoplasm or it can replicate in the nucleus. In a bacteriophage, it can only replicate in the cytoplasm. There are also differences in attachment and entry into the cell. It's much easier to get into an animal cell because an animal cell does not have a cell wall. How an animal virus gets into the cell is either by endocytosis, where the membrane invaginates around the virus, and then that'll be brought into the cell as a vesicle, and then the cell membrane will reform there. And there's the budding life cycle where the animal virus comes up to a membrane that has viral proteins in it, pushes on that membrane, creating a bud, and then rips off that bud. When it rips off that membrane then becomes the envelope to the enveloped virus. You should know that there's 7 viruses in the DNA family of viruses that cause human disease. And there's 15 RNA viruses that can infect humans and cause disease. 15 families, I should say. You should know the family Retroviridae is a little different, the retroviruses, because this is an RNA family of virus, and it's an exception to the rule of thumb where RNA virus families replicate in the cytoplasm, DNA family viruses replicate in the nucleus. The retroviruses are an RNA virus, but they replicate in the nucleus. Retroviruses has the enzyme reverse transcriptase. And did I mention that this is where we get the name retrovirus? Take the RE from reverse and the TR from transcriptase, and then you get the retro, sort of, except for the O, for retrovirus. Reverse transcriptase is a unique protein found in retroviruses. It is found in one other family of viruses, but mainly in retroviruses, and it allows DNA transcription from RNA. So the reverse transcriptase works on the RNA genome of this virus to make first single-strand DNA, and then the reverse transcriptase works on that single-strand DNA to make double-strand DNA. The double-strand DNA then integrates into the host cell's chromosome, and when that happens in any animal virus, certainly with retroviruses, but any other animal virus where the genome integrates into the host cell chromosome, that provirus, what it's called when the virus integrates into the host DNA, a provirus, will remain in that cell for the lifetime of the cell. The provirus will never come out. It will permanently become a genetic part of that cell for as long as that cell is alive. Any question about any of that? Now, for retroviruses, they can transcribe that viral genome, making the viral messenger RNA, which will then move into the cytoplasm to make the viral proteins. That viral messenger RNA is the genome of the virus, and so you get this genome of the virus together with the viral proteins, and then you get the virus starting to come up to a membrane, start budding out of it, ripping off a portion of that membrane, and then you get the retrovirus outside the cell. Any question about retroviruses? Coronaviruses are the virus that has COVID-19, and then viruses similar to COVID-19 like SARS and MERS. All three of these cause severe acute respiratory syndrome. Coronaviruses were well known to humans long before COVID-19 came around because it is the second most common cause of the human cold. Coronaviruses is the second most common cause of the human cold. The single positive strand RNA viruses, most people didn't know about it until COVID-19 came out, but we were briefly talking about it in microbiology before COVID-19 came out. COVID-19 is closely related to bat coronaviruses and likely originated from two different bat coronaviruses that recombine, and then those recombine with a pangolin coronavirus, so that three different viruses came about to make the first COVID-19, which then spread among humans. Unfortunately, COVID-19 has been changing. It tends to mutate about every 6 months. A new strain comes out. And one of those new strains came out was actually Omicron, which was a recombination between COVID-19 and a coronaviruses that caused the human cold. All subsequent variants of COVID-19 that have come out have been derivatives of Omicron. So all coronaviruses are now derivatives of, what is it? 2 bat coronaviruses, 1 pangolin coronavirus, and then a coronavirus causing the human cold. Any question about any of that? You know about viruses and cancer, that some cancers appear to be started by a virus. It's well known that cervical cancer, skin cancer, hepatitis, or liver cancer can be caused by certain viruses. Not all cancers are caused by viruses, though, like breast cancer and glioblastoma are not caused by a virus. And there's 2 ways that a virus can cause cancer. First, you can have, at least a human cancer, you can have the virus integrating into your genome, mucking up the DNA. It could mutate a gene, like a tumor suppressor gene, where the virus integrates right into the tumor suppressor gene. That tumor suppressor gene will not be made now. It could also turn on the expression of different genes, turn off the expression of different genes. And that's why I say muck up the DNA. And that is where the provirus integrates, it can change the expression of the nearby genes because of well, the DNA's integration, providing promoter sites, changing the regulation of the region around that gene. And then the second way that a virus can cause cancer is an oncogene. This is a virus containing a gene called an oncogene, which when this gene gets inside a cell, it causes the cell to grow uncontrollably. An oncogene is a normal cell gene involved in the growth of the cell, but this gene is mutated, and so the cell doesn't grow normally, it grows abnormally and then divides uncontrollably. Any question about the 2 ways that viruses can cause cancer? All right, you should know about latent and viral infection. I already talked about that. How acute infection, like the human cold, hits you rapidly, develops rapidly, then goes away rapidly. That's different from a latent infection where a child first gets chickenpox, the acute infection, and then the child many years later, maybe when they're an elderly adult, gets shingles. That's the latent infection. A persistent infection starts slowly, low numbers, tends to build up over time. It may bloom, it may kill the patient, or it may just bloom and then get better. Like mononucleosis is a persistent infection. It does bloom when the patient gets mono, but then they do get better and eventually even throw off mono. But HIV used to follow this, where the infection, the virus would grow, starting low, get worse, and then bloom when the patient gets AIDS, and then AIDS would kill the patient. You know about what prions are? A proteinaceous infectious particle where the protein infects a patient or animal and then causes disease by the protein replicating. How the protein replicates is it comes up to the normal protein, causing the normal protein to fold into the pattern of the prion protein. The prion protein and the normal protein have the same amino acid sequence, but they have a different folding pattern. All right, any question about any of that? Lastly, you should know that plant viruses cause certain plant diseases. And then viroids are a naked RNA molecule that causes disease. This potato plant were infected with a viroid, a naked RNA, which then reproduced in the cells, causing the tuber to be spindle shaped. This is a normal potato spindle tuber viroid, causing the potato to be this. All right, any question about plant viruses or viroids? You should know that viroids are only known in plant cells, and they are a similar thing, meaning it has a different name, but it's a viroid in a fungal. Those are the only cases we know of viroids. There are no viroids known in animal cells. All right, any question about chapter 13? All right, that is the review of the material since the last quiz. I will briefly continue in the lab with a review of the material with the quizzes, of the material that was on the quizzes. I'll be very brief about these topics, but I've now ran out of time, and so I want to end the lecture here, and that was a review of the material after the last quiz. Any question on anything? All right. I'll see you in the lab if you show up. If you don't,